Welcome to Gel Electrophoresis. We're going to go over the uses of this process and how this process works. To start, this process could be used to compare DNA left at a crime scene to the DNA of a suspect. So you could match DNA to find out who committed a crime or identify a victim of a crime. Second major purpose, you could find out who the father of a child is. Generally speaking, the mother knows their children. They came out of her. Every now and then, though, you do have to use this for the mother, just because sometimes babies might get mixed up in the maternity ward. But for the father, this is when it's mostly used. You compare the DNA of the mother and father to the children. And we'll take a look and see the ins and outs of that. Third big process or use is you could discover evolutionary relationships. You could see which organisms are closely related, which ones are more distantly related. And therefore you could fill out or complete a phylogenetic tree showing ancestry. Now let's take a look at how this process actually works. Alright, let's start by taking a look at how gel electrophoresis is used to catch a criminal. At a crime scene, there might be skin left behind from a criminal, usually underneath the victim's fingernails, as he or she is fighting back. It could be hair, it could be blood. All these tissues contain cells, because tissues are made up of cells. Skin, hair, and blood from one person will all contain the exact same DNA, because all the DNA of one individual, all the DNA in all their body cells are identical. So we took the crime scene DNA, we got suspect 1's DNA, suspect 2's DNA, and suspect 3 is being a bit of a pain. They don't want to give it up. So we follow them, we throw out a drink, we grab that straw or that cup, and we get some cheek cells off that cup or straw. Now we have 3 suspects' DNA and the crime scene DNA. The process of gel electrophoresis requires that it be cut up into pieces. Of course, this is going to be done by using an enzyme, but in particular, an enzyme that cuts up DNA called a restriction enzyme. It's called restriction because it only cuts, or is restricted to only cut, certain letter or base sequences, such as CCGG or AATT. And the same restriction enzyme is going to be used to cut up the crime scene DNA and suspect 1, 2's, and 3's. Because if the crime scene DNA is cut up into a bunch of pieces of specific lengths, and one of the suspect's DNA is cut up the exact same way, that means they have the exact same DNA sequence. You only have the exact same DNA sequence as a sample if that's your sample. All the cells in your body have the exact same DNA, but the cell or the DNA inside somebody else's cell, different. That's what makes us different, the letters in DNA. So if you use the same restriction enzyme to cut up the crime scene DNA, and all 3 suspects, whatever suspect has the same size pieces of DNA is guilty. All right. Let's see how the DNA is cut up. We have our restriction enzyme. It's going to cut wherever it sees the letters CCGG. So we mix it with the crime scene DNA. It's going to cut up the DNA like we've seen. But you know what? To make this simplified, we're just going to cut it right smack dab in the middle. So we have DNA is cut wherever CCGG is present, and the crime scene DNA is cut up into pieces. A piece of DNA that's 5 bases long, 10 bases, 11 bases, and 8 bases long. If one of the suspect's DNA is cut up into those same size pieces, though, that means that's their DNA that was left at the crime scene. Let's see who has the identical DNA. So suspect 1, let's put the restriction enzyme with that DNA. Again, it's the same one. It's going to cut wherever it sees CCGG. You get pieces of DNA that are 15, 13, and 6 bases long. Suspect 1's clear. Suspect 2. I could just tell you visually this is definitely not the crime scene DNA. You get pieces that are 27 bases long. Wow. 5 and 3. Suspect 3. Person who did not want to give their DNA up. The DNA is cut into pieces that are 5, 10, 11, and 8 bases long. That's the exact same cuts that were made in the crime scene DNA. Since they were the exact same cuts, they make the exact same size pieces. But there's a problem. We can't just look under a microscope and count the letters on DNA. Therefore, we have to find a way to compare these cut up, different size pieces of DNA. And for that, we need a gel. So we're going to take the cut up DNA, show where it is in the picture. So our cut up DNA, we have our cut up DNA now. And we're going to put it into the gel. Because again, we can't sit under the microscope counting letters. So this gel will help separate the DNA based on what size it was cut up into. Therefore, the gel is filled with microscopic pores or holes so the DNA can move through. At the beginning of this gel, or at one end, there are little wells. This is where those cut up DNA samples are going to be put. A little micropipette is used to squirt in the DNA. Suspect 1's DNA, 2's DNA, and 3's DNA are all put into these wells. Taking a different look or a closer look at the wells, on the left you see the DNA going in. On the right you see the DNA going in at a different angle. It's going into these little wells. Back to our picture. The DNA is now in the wells. The wells are inside a gel filled with holes. The DNA wants to go through those holes and move to the other end of the gel, but we need to see what causes it to move. So let me just focus on that point that I just made. You have cut up pieces of DNA at one end of the gel. Big pieces, smaller pieces, varying size pieces. And we said this gel is filled with holes. So when the DNA moves through, these tiny little pieces will go super far because they're able to snake their way through and move through the holes in the gel. And as you get bigger, the bigger the piece, the less far they travel. So small pieces travel super far. These big pieces can't travel as far. Oops. And this gigantic piece goes like nowhere. And you're going to have a lot of those pieces build up, forming distinct DNA bands, or bands of DNA. And it's going to leave a very unique pattern for each individual. So you could think of this as like your DNA fingerprint. It's unique to you, a DNA banding pattern, or if you really want to go stretch out the words DNA barcode. So barcodes are unique, so is this. But use the words DNA banding pattern or DNA bands. That's ideal. Just a reminder, the smallest travel farthest, the longest don't travel far at all. They stay close to the wells. So the cut up DNA is in the wells in the gel that's filled with holes, and electricity or an electric current is going to cause the DNA to move, because DNA has a negative charge. And if you see, the wells have the negative end. Negative and negative don't want to stay together, so the negative DNA wants to go to the positive end. The smallest pieces of DNA will travel through the holes best and go farthest. The largest pieces of DNA will stay up close. And now we can compare the DNA bands. And we see that the DNA bands all the way on the left match the DNA bands all the way on the right. Exact same DNA pattern. That means suspect 3's DNA has to be identical to the crime scene DNA all the way to the left. Meaning they have the same exact sequence of letters, therefore it was cut up in the exact same spots, making the exact same size pieces, creating the same banding pattern. So let's take a look at our scenario, but from scratch. Who's guilty in this scenario? Suspect 1, 2, or 3? Well, you take the crime scene DNA bands and you try and see who has the exact same bands. Mind you, all the bands have to match. If there's 1 or 2 bands present in the crime scene DNA but not in, let's say, suspect 3, suspect 3 didn't do it. But in this case, suspect 3, their DNA banding pattern is a complete match. Exactly the same. That means that all their DNA bands match. They must have the exact same DNA, because it was cut up into the exact same size pieces. And you cannot say it has most of the DNA bands in common. Well, I don't want to go to prison for the rest of my life if, oh, most of them are right. No, it has to be a complete match. Here's a new scenario. We have a female and a cub, but there are 2 males. We're not sure which male the father is. So, in order to find out which one the father is, we have to use gel electrophoresis, cut up DNA into different size pieces, and then compare those pieces of DNA. But in order to make sense of this process, you have to know an important thing. So in order to understand this fully, we have to look at a sperm and an egg. Sperm and egg sperm has half dad's information, egg has half mom's DNA information. Together, half your dad's DNA and half your mom's DNA makes you a full diploid cell, a zygote. Technically, you are 100%, actually, yeah, a wrong time to use that. You are 50% your mom, half your mom, and you are 50% your dad, half your dad, genetically. So you have half your dad's DNA and half your mom's DNA. All your DNA comes from mom or dad. Back to our little picture here. We see the cub. It has very distinct bands of DNA. All the DNA must be accounted for by mom and dad. So these pieces of DNA all came from mom. But there is one DNA band, a larger band, that must have came from dad because it did not come from mom. Therefore, let's see, male 1 is the only one who could have given the cub that chunk of DNA. Male 2 does not have that same identical band. Therefore, male 1. All the DNA bands that did not come from mom, all of them came from dad. In this case, male 1. Not most of them, all the bands. In our second story here, we got a mom, a child, 2 males. Not sure which one the dad is. So which male tiger is the father of the newborn cub? Explain. We're going to start off the same. We're going to take all of mom and the baby's DNA and match them up. So all these bands came from mom, and these green things I'm highlighting, these must have came from dad. It's a lot. So let's look at these green highlighted bands and match them up to either male 1 or male 2, and they all have to match up, not just most. So these blue bands, it looks like some of them are inconclusive. But here, all the DNA bands that are present in the child, all those DNA bands that did not come from mom, the one circled in red, all came from dad, the one circled in blue. So again, that is definitive confirmation that male 2 is the father. All right, new scenario. Parents 3 and 4 are in the middle. Which puppy might have been placed in this litter by mistake? We're going to look for the puppy, either 1, 2, 5, or 6, that has bands of DNA that don't match parents 3 or 4. Because if the bands don't match parents 3 or 4, that means the parents 3 and 4 did not give those bands of DNA to you. So we just have to find the puppy that have DNA bands that don't match mom or dad. Puppy 1, they have bands from mom and dad. Puppy 2, these 2, they don't match mom or dad. That was kind of quick. It looks like these DNA bands must have come from a different mom and dad. Puppy 2, therefore, is definitely not part of this family genetically, because it has no DNA bands from either mom or dad. And we could do 5 and 6. You could see puppy 5 has the first DNA band from parent 4, the second one's from 3, and the other puppy, puppy 6, the 2 bands are from mom and dad. Last scenario. Which 2 organisms are most closely related? It seems like a crazy question, a long-winded one, but you have to compare all the organisms. So I just kind of start with going, organism 1 compared to organism 2, 1 to 3, 1 to 4, and then 2 to 3, 2 to 4, and then 3 to 4. I'm going to compare and see how many DNA bands they have in common. If you have a lot of DNA bands in common, you're very closely related, and you probably have a very recent common ancestor. So organism 1 and 2, they only have 1 band in common. 1 and 3, 1 band in common. 1 and 4 have 2 bands in common. Ooh. Organism 2 and 3, 1, 2, 3 bands in common. Wow. 2 and 4, 2 bands in common. 3 and 4, 1. Organisms 2 and 3, they are the most closely related, the most genetically similar, therefore probably share a very recent common ancestor. Just being a little thorough here with that answer. And it's fine to use the most here because you're just finding which one's most closely related, not a definitive who's the dad or who's the criminal. Some practice questions or review questions. Gel electrophoresis is used for what? Compare crime scene DNA to suspect DNA. Find out who committed a crime. 2, find out who the father is, or who a parent is, or which child doesn't belong genetically to a family. Or 3, you find closely related species. See which species is more closely related to a human or more distantly related to a human. A lot of implications behind that. What's special about the gel? It's filled with microscopic holes or pores so the DNA is able to snake its way through. Electricity is the main vehicle or motor behind moving DNA. DNA is separated based on its size. Really tiny pieces go super far. Very large pieces stay very close to the wells. It doesn't travel much through the gel because it's so big it can't fit through those holes. This next question, question E, little spoiled. Kind of just said the answer. The smallest pieces are able to snake their way through the holes and go very far. And the largest pieces can't travel that far because they're very big. So they're going to stay very close to the wells. Letter F, what's used to cut up DNA? Restriction enzymes. And there's a video, a separate video, like a 4-minute one, that just goes over that real quick if you're looking to take a deeper look at restriction enzymes. This last question, G, if someone leaves a piece of hair, skin, and blood behind, will they all lead back to that one person? Yep. Because all your cells in your body have the exact same DNA. Minus your gametes, of course, they have half your DNA. But all your body cells have the exact same DNA because they were all made by mitosis. Where did all your cells come from? The zygote. You were one cell big at one point in time, and all the trillions of cells that make up your body came from that one cell with that DNA. They all came from that cell by means of mitosis, which makes identical cells. But cells start to use different genes at a certain point in development, allowing them to carry out different functions and make different organs. This is called cell differentiation. It's when cells start to become specialized. Thank you for watching. Stay tuned for more.